So let's talk about T cells or cell-mediated immunity. They're a little different because they're differentiated in the thymus and they have a different line of attack that they use. B cells basically make antibodies to tag things, so they directly don't get dirty. They basically tag them with antibodies, and the antibodies go ahead and just flag white blood cells to come and kill them. But T cells, they're a little different. They go ahead and do the work for us, or they get other people to come and help us. So the T cells, they start to differentiate in the thymus, but they hang out in your lymphatic system. The T cells aren't really recognized antigens, but what happens is when they come in this MHC complex that we're going to talk about in a minute, then they can go ahead and recognize the item and start doing whatever they need to do. And this one's going to use this process called MHC1. So what happens is there's different types of T cells, there's different subclasses, and we'll talk about each. There's the cytotoxic T cell, or the TC cell. If you're taking the MCAT, you probably want to know them as CD8s. So these carry out, they have the molecules on the surface. So what happens is we have the MHC1 complex that comes in and recognizes whatever foreign body it is, comes over and it presents the antigen through the MSC1 complex to the T cytotoxic T cell. And the cytotoxic T cell is also known as a killer cell because what it does is it will make these cytotoxins. One of them is called perforins, and perforins are this chemical that they'll actually put in that cell, and it makes holes in it. So perforin almost sounds like perforate, and perforate means put holes in something. It also can induce this thing called apoptosis, which is programmed cell death. Sometimes what happens is some of our cells get sick or they have cancer and they can't kill themselves. It's an auto-destruct mechanism cells have, and it's called apoptosis, just programmed cell death. So what will happen is the cytotoxic C cell will come and hit it with chemicals, and it'll cause the cell to sort of die. And it sounds terrible, but if your body didn't do this process, what would happen is you would start to have all these old cells that would start to make mistakes, and you would have died of cancer probably in year 1 or year 2. Our next class of T cells are going to be our T helper cells, or TH cells. If you're taking the MCAT, they're called CD4s. They go ahead and they recognize antigens on that MHC2 complex. Remember how we talked about the TC cell recognizing MHC1? Well, this one recognizes MHC2, and what happens is they are not really designed to kill, but what they do is they stimulate the immune system of the cell-mediated. So that's going to be the cytotoxic T cells, the NK cells, which we'll talk about in a minute, the natural killer cells. They'll also help with the macrophages. Those things all come around and just eat up foreign bodies. It also stimulates the production of eosinophils if they're needed, and the antibodies IgM and IgE. Then we have these regulatory cells, and what they're designed to do is when everything sort of is done, they go ahead and turn everything down back to normal. If not, then your body would continue to ramp up, and never sort of stop the attack, even though nothing was around anymore. So these guys go ahead and tell everybody to sort of chill out, we're done. Our last class of T cells are these ones called natural killer cells. And what natural killer cells do, they act similar to the cytotoxic T cells, except with the cytotoxic T cell, they need an antigen present on the outside to do its job. This guy will go ahead and kill your cells, the ones that aren't behaving correctly or doing something correctly. And what they're really trying to do is take them out, and these are going to be viruses, cells that have been infected by viruses, not the virus itself, but cells infected with viruses, cells that seem like they might be turning into cancer or tumors themselves, and then anything that has to do with parasites, they'll take them out too. So they don't really need the whole presentation thing. They're just looking for cells that don't look like they're looking right or doing their functions correctly, and they kill them, or parasites. So let's talk about the sequence of events of how B cells and T cells interact and work together. So before we even do that, let's talk about the differences between B cells and C cells. So B cells differentiate in the bone marrow. T cells differentiate in the thymus. B cells are uneducated. They're kind of like bonehead cells. They don't really they don't have the MHC process like with 2. The T helper cells, educated cells, they recognize MHC2, but they don't attack. The only response is they only present or work if there's an antigen present, unless we're talking about the natural killer cells or the T regulatory cells. But really, here's the big picture. The big picture is that let's focus on this step first, which is the TC cell. This is what I'm kind of hoping that you regurgitate for the exam. So we have some foreign bodies, and they're picked up and sort of regurgitated in this cell. And this cell is going to go ahead and do this complex called MHC1, where it takes a little part of this and presents it to the outside. When this happens, what happens is that cytotoxic T cell is going to come over and touch it. And what it'll do is it'll release perforins. And when it releases perforins, what happens is the cell will start to implode and die. So without the MHC1 complex, this wouldn't happen. And this is just with TC cell. If we look at our second process down here, we have our foreign microbes come in, and MHC2 is presented. So MHC2 is really designed for the T helper cell. And what the T helper cell is going to do is they're going to go ahead and, number one, they want to kill off anything that might be in here still alive. So the TC cell, the cytotoxic T cell, comes and will kill the cell off. The other thing that this T helper cell will do is it's going to go ahead and inform the B cells. The B cells will go ahead and start cloning themselves. And when they clone themselves, they're going to become either plasma cells or some will be memory cells. Remember, the plasma cells will become those antibodies and sort of attach to these foreign things and flag them. And the memory cells are going to hang out for a while, and then eventually, when the fight is completely done, sort of turn off the fight and sort of, go back into normal stasis. The B cells alone could work on their own. They don't need any of this process. But basically, when they do that, they're sort of working alone. Their plasma cells are tagging these things, and that's really it. And the white blood cells are coming and killing them. But to get the complete kill, we want to go ahead and get all of these involved. And the only way we can get the whole specific immune system involved is if we get the T helper cell. If the T helper cell wasn't there or got taken out, what would happen is this would be one arm of your immune system, this would be one arm, but they wouldn't work together. This guy is like the general. He kind of coordinates everything and makes sure that you can kill everything at one time. Great. I think that's all I wanted to talk about with these two different types of immune systems.